Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Gram-positive cocci made easy, Staphylococcus and Streptococcus. Medical Microbiology Lecture. Table of Content. Gram-positive cocci of medical importance. Staphylococci, Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus, Streptococci. Group A Streptococcus, Streptococcus Pyogenes. Group B Streptococcus, Streptococcus Agalactiae. Group C and D of Streptococci. Group D Streptococci, enterococci and non-enterococci. A-Hemolytic Streptococci, Viridans Group. Streptococcus pneumoniae, the pneumococcus. Streptococci. General characteristics of Streptococci. Shape and arrangement. Gram-positive spherical Dodgic ovoid cocci, usually in long chains, may also appear in pairs. Spore and motility, non-spore-forming, non-motile. Surface structures. Some species can form capsules and slime layers. Oxygen requirement. Facultative anaerobes. Enzymes. Lack catalase, but possess a peroxidase system, distinguishes them from Staphylococci. Colony morphology. Produce small, non-pigmented colonies. Environmental sensitivity. Relatively sensitive to drying, heat, and disinfectants. Gram reaction. Variability. Although gram-positive, older cultures may lose gram positivity and appear gram-negative, especially after overnight incubation. Classification of Streptococci. Classification of Streptococci is based on several criteria observed over time. Colony morphology and hemolytic reactions on blood augur help in differentiation. Serologic specificity of the cell wall group, specific substance, Lancefield antigens, and other capsular or cell wall antigens is a major basis. Biochemical reactions and resistance to various physical and chemical factors provide further classification. Ecologic features, including natural habitat and pathogenic potential, are also considered. Hemolysis of Streptococci. Many Streptococci can hemolyze red blood cells in vitro to different extents. Beta hemolysis, complete lysis of erythrocytes, producing a clear zone around colonies. Alpha hemolysis, partial lysis of erythrocytes with hemoglobin reduction, giving a greenish discoloration. Gamma hemolysis, non-hemolytic. No hemolysis observed. Beta-hemolytic streptococci are also called pyogenic streptococci, which include important human pathogens such as Streptococcus, Pyogenes, Streptococcus Agalactiae, and Streptococcus dysgalactiae. Lancefield classification. Lancefield classification system is based on cell wall antigens, dividing Streptococci into 20 serologic groups, A to H, K to U. The group-specific carbohydrate antigen determines the serologic specificity and is composed of an amino sugar, Group A Streptococci, rhamnose, N-acetylglucosamine, Group B Streptococci, rhamnose glucosamine polysaccharide, Group C Streptococci, rhamnose, N-acetylgalactosamine, Group D Streptococci, glycerol tioic acid containing D-alanine and glucose. Group F Streptococci, glucopyranosyl N-acetylgalactosamine. Capsular polysaccharides of Streptococci. The antigenic properties of capsular polysaccharides are important for classification. Streptococcus pneumonia is divided into more than 90 distinct types based on its capsule, while the capsule also serves as the basis for typing group B Streptococci, Streptococcus Agalactiae. Biochemical reactions of Streptococci. Streptococci can be differentiated using biochemical tests such as sugar fermentation profiles, enzyme detection tests, susceptibility or resistance to specific chemical agents. These tests are typically applied after assessing colony morphology and hemolytic patterns. Human streptococcal pathogens. Streptococcus pyogenes, Streptococcus Agalactiae, Viridans Streptococci, Streptococcus pneumoniae, Enterococcus faecalis. Epidemiology and Pathogenesis of Streptococci. Humans serve as the sole reservoir, with inapparent carriers playing a key role in transmission. Spread occurs through direct contact, respiratory droplets, contaminated food, and fomites. Entry usually occurs via the skin or pharynx. Infection patterns vary with environmental conditions. Skin infections, common in warm, humid climates. Pharyngeal infections, frequent in winter and dry conditions. Children are most often affected by cutaneous and throat infections. If untreated, systemic infections and serious complications may develop. Group A Streptococcus, Streptococcus pyogenes. Considered the most significant pathogenic Streptococcus, exists as a strict parasite, commonly resides in the throat, nasopharynx, and occasionally on the skin. Colony morphology of Streptococcus pyogenes. Cells are spherical or ovoid cocci arranged in chains. Streptococcus pyogenes shows beta hemolysis. Cultural characteristics of Streptococcus pyogenes. Streptococci generally form discoid colonies on solid media, about 1 to 2 millimeters in diameter. Growth and hemolysis of Streptococcus pyogenes are enhanced when incubated in 10% CO2. Variation of Streptococcus pyogenes. A single strain of Streptococcus may produce colonies with different appearances. In Streptococcus pyogenes, 2 colony types are common, mat colonies. Produce abundant M protein, usually associated with virulence. Glossy colonies. Produce little M protein, often a virulent. Virulence factors of Streptococcus pyogenes. C carbohydrates protect the bacteria from lysozyme action. Fimbriae aid in bacterial adherence to host tissues. M protein provides resistance against phagocytosis, a key virulence determinant. Hyaluronic acid capsule non-immunogenic, helps bacteria evade immune recognition. C5 a protease disrupts complement activity and inhibits neutrophil response. Toxins and enzymes. Streptolysins, SLO and SLS. Hemolysins that damage red blood cells and various other host cells. Erythrogenic, pyrogenic toxin. Causes fever and the characteristic red rash of scarlet fever. Superantigens. Overstimulate T cells by binding to MHC class II and T cell receptors, leading to massive cytokine release, shock, and tissue damage, similar to staphylococcal toxic shock syndrome toxin. Extracellular enzymes, streptokinase, converts plasminogen to plasmin, breaking down fibrin clots and aiding bacterial spread. Antibodies, Anti-streptokinase may develop after infection, also used therapeutically in clot treatment. Hyaluronidase breaks down hyaluronic acid in connective tissue, facilitating spread. Antigenic and induces specific antibodies. Streptodornase, DNase A, B, C, D, hydrolyzes DNA, reduces pus viscosity, and helps bacteria spread in tissue. Antibodies to DNase, especially DNase B, are diagnostic after skin infections. Clinical findings of Streptococcus pyogenes. Invasive infections. The portal of entry determines the clinical manifestation. Infection spreads rapidly along lymphatics and may enter the bloodstream. Erysipelas, entry via skin, causes massive brawny edema with a sharply raised red advancing margin of infection. Cellulitis, acute, rapidly spreading infection of skin and subcutaneous tissues after trauma, burns, wounds, or surgery, characterized by pain swelling, tenderness, and erythema. Unlike erysipelas, lesions are not raised, and margins are indistinct. Necrotizing fasciitis, streptococcal gangrene, severe infection of subcutaneous tissue with rapid necrosis of skin and underlying layers, often termed flesh-eating bacteria. Puerperal fever, infection enters the uterus after delivery, causing septicemia originating in endometritis. Bacteremia, sepsis, may follow traumatic or surgical wound infections, cellulitis, or rarely pharyngitis, rapidly progressive and potentially fatal. Localized infections. Streptococcal sore throat, pharyngitis. Most common β-Hemolytic Streptococcus pyogenes infection. Bacteria adhere via lipoteicoic acid-covered pili and hyaluronic acid capsule. Characterized by acute nasopharyngitis, tonsillitis, intense redness, and edema of mucous membranes. Does not usually involve the lungs. Streptococcal pyoderma, impetigo, superficial skin infection in children, presents as vesicles that rupture and form pus-covered erosions later encrusted. Highly communicable, spreads by contact, and is common in warm, humid climates. Systemic invasive infections. Scarlet fever. Follows throat infection when strains produce pyrogenic toxin. Toxin spread causes high fever and a diffuse bright red rash over face, trunk, and extremities. Streptococcal toxic shock syndrome. Severe invasive infection leading to shock, bacteremia, respiratory failure, and multi-organ failure. Mortality is approximately 30%. Often follows minor trauma in otherwise healthy individuals, usually associated with soft tissue infections. Epidemiology and Pathogenesis of Streptococcus pyogenes. Humans serve as the only natural reservoir. Carriers may remain asymptomatic. Spread occurs through direct contact, respiratory droplets, contaminated food, or fomites. Usual portals of entry are the skin or pharynx. Type and frequency of infection vary with climate, season, and living conditions. Skin infections are more common in warm, humid environments. Pharyngeal infections occur more often in colder, dry climates, especially during winter. Children are the main group affected by both skin and throat infections. If left untreated, systemic infections and serious sequelae can develop. Post-streptococcal diseases. Post-streptococcal diseases occur after an acute Streptococcus pyogenes infection, leading to nephritis or rheumatic fever. Latent period, 1 to 4 weeks. These diseases are not caused by direct bacterial effects, but represent a hypersensitivity response. Nephritis is more commonly preceded by skin infections. Rheumatic fever is more commonly preceded by respiratory tract infections. Rheumatic fever, causes damage to heart muscle and valves. Presents with fever, malaise, migratory polyarthritis, and inflammation of all parts of the heart. Acute glomerulonephritis, initiated by antigen-antibody complexes deposited on the glomerular basement membrane. Clinical features of acute nephritis: blood and protein in urine, edema, high blood pressure, urea nitrogen retention, and low serum complement levels. Group B Streptococcus, Streptococcus Agalactiae. Group B Streptococcus Agalactiae is part of the normal flora of the human vagina, pharynx, and large intestine. It can be transferred to infants during delivery and cause severe infections. It is the most prevalent cause of neonatal pneumonia, sepsis, and meningitis. Pregnant women should be screened and treated to prevent transmission and infection in newborns. It can also cause wound infections, skin infections, and endocarditis in people who are at risk. These bacteria are typically β-hemolytic and produce zones of hemolysis that are only slightly larger than the colonies, usually about 1 to 2 millimeters in diameter. Group B Streptococci can hydrolyze sodium hippurate and give a positive response in the CAMP test. Christie, Atkins, Munch-Peterson. CAMP test. CAMP test. Group B Streptococci, Streptococcus agalactiae, when streaked perpendicular to Staphylococcus aureus, produce CAMP factor. The CAMP factor enhances the hemolytic activity of Staphylococcus aureus, creating a distinct arrow-shaped zone of hemolysis. Streptococcal tests. Streptococcal infections pose the highest risk to elderly and immunocompromised populations. Predisposing factors include diabetes mellitus, cancer, advanced age, liver cirrhosis, corticosteroid therapy, HIV infection, and other immunocompromised conditions. Major clinical manifestations, in descending order of frequency, are bacteremia, skin and soft tissue infections, respiratory infections, genitourinary infections. Diagnostic laboratory tests of Streptococcus. First specimens. Type of specimen depends on the site of streptococcal infection. Common specimens: throat swab, pus, blood for culture. Serum is collected for antibody detection. Second smears. Smears from pus often show single cocci or pairs, not distinct chains. Sometimes appear gram-negative due to non-viability and loss of ability to retain crystal violet. Third culture. Suspected samples are cultured on blood agar plates. Incubation in 10%. CO2 enhances hemolysis. Blood cultures may yield hemolytic group A streptococci, e.g. in sepsis, within hours to a few days. Some non-hemolytic streptococci and enterococci grow slowly. Hemolysis pattern. Group A streptococci may be presumptively identified by growth inhibition with bacitracin, though more reliable confirmatory tests are preferred. Fourth antigen detection tests, used for rapid identification of Group A streptococci. Fifth serologic tests, detection based on rise in antibody titers to Group A streptococcal antigens. Important antibodies include ASO, anti-streptolysin O, widely used, especially in respiratory infections, anti-DNase and anti-hyaluronidase, useful in skin infections, anti-streptokinase, anti-M-type specific antibodies, and others. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Group C and D of Streptococci. Group C Streptococci infrequently cause human infections. Group D Streptococci classified into enterococci and non-enterococci. Enterococci include Enterococcus faecalis, Enterococcus faecium, Enterococcus durans. Normal colonizers of the human large intestine cause opportunistic urinary, wound, and skin infections. Can grow in 6.5% salt solution or bile and are not killed by penicillin G, usually non-hemolytic, but occasionally α-Hemolytic. Group D Streptococci, enterococci, and non-enterococci. Non-enterococci, Group D Streptococci. Non-enterococci include Streptococcus bovis, sensitive to 6.5% salt concentration and killed by penicillin. G show variable hemolytic patterns. Identification is essential for proper treatment and to prevent complications. Rapid diagnostic tests use monoclonal antibodies reacting with C carbohydrates. Culture methods include bacitracin disc test, CAMP test, esculin hydrolysis. Treatment of Streptococcus. Groups A and B Streptococci are treated with penicillin and amoxicillin. Long-term penicillin therapy is required for patients with a history of rheumatic fever or recurrent strep throat. Enterococcal infections usually require combined therapy, e.g., penicillin or ampicillin with aminoglycosides. A-Hemolytic Streptococci, Viridans Group. Large complex group includes Streptococcus mutans, Streptococcus oralis, Streptococcus salivarius. Growth not inhibited by opticin, and colonies are not soluble in bile deoxycholate. Most numerous and widespread residents of gums, teeth, oral cavity, and also found in nasopharynx, genital tract, and skin. Not highly invasive, but dental or surgical procedures facilitate entry into the bloodstream. Cause bacteremia, meningitis, abdominal infections, and tooth abscesses. Most serious infection is subacute endocarditis. Blood-borne bacteria settle and grow on heart lining or valves. Persons with pre-existing heart disease are at high risk. Colonization of the heart occurs via biofilm formation. Streptococcus mutans produce slime layers that adhere to teeth, forming the basis of plaque. Involved in dental caries. Persons with pre-existing heart conditions should receive prophylactic antibiotics before surgery or dental procedures. Streptococcus pneumoniae, the pneumococcus, causes 60 to 70% of all bacterial pneumonias. Cells are small, lancet-shaped, arranged in pairs or short chains, possess a polysaccharide capsule, enabling typing with specific antisera. Lack catalase and peroxidases. Cultures die in the presence of O2. Cultural characteristics of Streptococcus pneumonia. Colonies are small, round, dome-shaped initially, later developing a central plateau with an elevated rim. A-Hemolytic on blood agar. Growth is enhanced by 5 to 10% CO2. Growth characteristics of Streptococcus pneumonia. Obtain most energy from glucose fermentation. Rapid production of lactic acid limits growth. Virulence factors of Streptococcus pneumonia. Disease results from ability to multiply in tissues. Pneumolysin, cytolytic and inflammatory toxin. Capsule, major virulence factor that prevents, delays phagocytosis. 90 capsular types identified. Immunity is type-specific. Serum antibodies to capsular polysaccharide provide protection. Immunization with a given capsular polysaccharide confers immunity against that type. Encapsulated strains are at least 10 to the power of 5 times more virulent than unencapsulated strains. Cell wall and cell wall polysaccharide of Streptococcus pneumonia. Unlike capsule polysaccharide, peptidoglycan and cell wall polysaccharide can independently induce inflammatory responses similar to whole pneumococcal infection. Pneumococcal proteins of Streptococcus pneumonia. Immunoglobulin A1 protease interferes with host mucosal defenses. Neuraminidase may aid epithelial attachment by cleaving host sialic acid. Pneumococcal surface protein A shows structural and antigenic variability across strains, present in most clinical isolates. Epidemiology and Pathology of Streptococcus pneumonia. Carriage rate. 5 to 50% of people harbor it as normal. Nasopharyngeal flora. Infections are usually endogenous, very delicate, does not survive long outside host. Risk groups: young children, elderly, immunocompromised, patients with lung disease, viral infections, and those in close quarters. Pneumonia occurs when bacteria are aspirated into lungs of susceptible hosts, leads to overwhelming inflammatory response. Can also spread to middle ear via the eustachian tube. Pathogenesis of pneumococcal disease. Colonization. Streptococcus pneumonia is commonly carried in the upper respiratory tract of healthy individuals. Attachment to host cells is suggested to be mediated by a disaccharide receptor on fibronectin present on human pharyngeal epithelial cells. Adherence and predisposing factors. Pneumococci adhere to tracheal epithelial cells, and this adherence is enhanced by prior influenza virus infection, increasing susceptibility. Invasion and spread. Mechanisms of translocation from the nasopharynx to the lung, causing pneumonia, or directly into the bloodstream, causing bacteremia, septicemia, are not fully understood. Bacterial multiplication and lysis. Uncontrolled multiplication in the lungs, meninges, or middle ear leads to pneumococcal lysis. Lysis releases cell wall components and pneumolysin, both of which are key in triggering host responses. Lysozyme secretions may contribute by activating autolysin, further promoting bacterial lysis. Inflammatory response. Pneumococcal lysis induces strong inflammation. Directly, by attracting and activating phagocytes. Indirectly, through complement activation. This excessive inflammatory response is believed to be a major factor responsible for morbidity and mortality in pneumococcal infections. Clinical findings of Streptococcus pneumonia. Sudden onset with fever, chills, and sharp pleural pain. Sputum, bloody or rusty colored. Spontaneous recovery may occur in 5 to 10 days. Also causes otitis media, sinusitis, and purulent bronchitis. Diagnosis of Streptococcus pneumonia. Gram stain, presumptive identification. Quellung test, capsular swelling reaction. Culture characteristics. α-Hemolytic, opticin sensitive, bile soluble, inulin fermentation. Treatment and prevention of Streptococcus pneumonia. Traditionally treated with penicillin, G or V. Drug resistance increasingly reported. Capsular antigen vaccine for older adults and high-risk individuals, effective for 5 years. Conjugate vaccine for children 2 to 23 months. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!